Good afternoon, and welcome to Energene's webinar series. My name is Yael, and I am part of the marketing team, and I'll be your host for today. In today's webinar, we'll discuss the cannabis genome and the challenges of working with the under-researched crop. We'll also learn about QIAgene's breeding project done in collaboration with Energene and present some interesting results for trait discovery that focuses on the autoflowering trait. At the end of the webinar, we will save some time for questions, so we encourage you to send in your questions at any time during the presentation by using the Questions tab on the left side of your screen. This webinar is being recorded and will be posted on our website in case you'll want to listen to it again. It's my pleasure to present today's speakers. Our first speaker is Dr. Christopher Hone. Chris brings over 12 years of experience in plant breeding and genetics to drive innovation, breeding technologies, and research at QIAgene. As the Director of R&D, he is leading QIAgene to the forefront of cannabis breeding and developing varieties that are stable, productive, and tailored to the farmer's needs. I would also like to welcome Dr. Barak Marcus. Barak is a senior computational biologist at Energene. Over the years, Barak gained extensive experience in algorithm development in both academia and the industry. Specializing in population genetics of human and plant genomics, he is now leading the development of genetic mapping methodologies at Energene. We will now begin. Chris, the stage is yours.
Thank you. Thank you everyone for joining us today. I want to start out by introducing you guys to QIAgene. So we're a genetics company, and we're leveraging breeding technologies like those that we're going to discuss today to pioneer cannabis breeding. And we were focusing on delivering stable hemp varieties that are relevant to the market. And we're building that success by utilizing our vast experience coming out of the seed industry world. Okay, so our program in general, from a high level, looks like this. It begins with germplasm discovery, trait discovery, and development. So we identify germplasm that has traits that we're interested in incorporating into our breeding program. We then introduce these traits to our breeding lines through targeted selection and generation advancement. And those breeding lines are then used in our heterotic crossing blocks to generate hybrid crosses that we evaluate in a multi-year, multi-location system with university collaborators and grower collaborators. And if any of those varieties check all our boxes, meet our criteria, then they get commercialized and sold to farmers. And through strategic collaborations like the one we're talking about today with Energene, who are leaders in their field of genomics, we're able to develop technology and data to deliver stable varieties that are cannabinoid rich and can be used for a range of therapeutic and industrial applications. And a big portion of our program is the trial evaluation period. We believe that it's important to trial a lot so we can understand our varieties before they go out into the grower's field. So the grower's not learning about the variety when they're just getting it in the field. We want to be able to have answers and be able to share with them as much information as possible. So we're very data driven, which can help instill confidence into our customers. And down here I have a general life cycle of what a hybrid goes through. Initially we have our phase one, our internal trials, where we evaluate performance. We look at things like compliance, market acceptability, and then if it fits the portfolio that we were breeding for. If it meets those, then the top performers go on to phase two, which is our external trials, where we work with key collaborators in regions across the US to get these into different environments. So we again look at compliance, adaptability across those locations, and overall performance across the locations. And then the top performers move on to the third phase, which is commercial trials. These are larger trials with university collaborators, grower collaborators up to an acre in size. We look at overall performance again, yield, and then the marketability of the product coming from those varieties. And if it meets all the criteria, then we will commercialize it into a new variety. So our history with Energene goes back to October 2018, not too long after Kyogene was formed as a company. And we approached Energene with the intent to improve cannabis breeding and seed production and develop the tools to enable us to be really good at that. And that began with a handful of genomes that we were going to sequence, and Energene was going to assemble and deliver fully phased genomes to us. So in February 2019, not too long after we started, we had our first fully phased cannabis genome, and this was just the first of many. We continued on to do more to build a good foundation for our breeding program. And in March 2019, utilizing 2 of the genomes that we had sequenced and assembled with Energene, we began the project that we're talking about today, which is the Autoflower QTL mapping project. And we have some very good results that we'll be sharing with you, and excited about that. In October of last year, we began utilizing Energene's CanScan offering, which looks at germplasm diversity. So we want to look at the diversity within our germplasm collection, understand our heterotic groups so that we can better design our hybrid crosses and better use our germplasm moving forward as we're breeding. So for this year and beyond, we're currently discussing with Energene about utilizing their CanaGene database to create a custom database, which will really enable us to use the data that we've been generating. And it fits right in line because we've gone through kind of all their offerings, from de novo magic, the genome assembly, to trait magic QTL mapping, which is the Autoflower project we're talking about, CanScan for our diversity, and then now CanaGene to put it all into a database that we can make use of it. So for the project that we're talking about today, there were some challenges in getting started. One, like I mentioned, Kyogene was a young company. Our breeding program was young, so we had heterozygous parent materials that we were working with. They were fixed for the autoflower trait, but they were heterozygous. And being that autoflower plants are generally small and have a short vegetative period, it was impossible to collect enough tissue from a single plant to get to be able to do the 30x sequencing. So we had to come up with a clever solution. So by putting together our heads with Energene, we consulted with them, and we came up with this pedigree which could work around these challenges. So utilizing 2 of the parent lines that we had full sequence, fully phased genomes for, represented here by the 2 squares, the blue and the green, we were able to cross those to the same autoflower parent to generate the F1 progeny, which we would do the 30x sequencing on. And then we would select which of those F1s had the phenotypes that we wanted, cross those to generate 200 progeny that we would do 1x sequencing on. And the beauty of this design was that by using the same autoflower parent and having the full genomic information on the 2 parent lines that we used, we could then infer and decipher which haplotype blocks in the F1 and the progeny came from that autoflower parent. So this process took a bit of time. Typically from seed to seed, it's a 4-month period. So this first cross took about 4 months. In the second cycle, we had to first phenotype. So we had to grow out these photoperiod plants, select which ones had the contrasting phenotypes so that we made sure the progeny would segregate for the other phenotypes besides the autoflower trait that we were interested in, and then clone those so we could have enough tissue to collect for the sequencing, make the crosses, get the seed of the progeny. And then the phenotyping of the progeny had to be done in 2 steps as well. So we had to grow out the progeny under 18 hours of light initially so that we could identify the autoflower progeny. Once those were identified, we then cloned the photoperiod progeny, flowered out those clones, collected the phenotype data for other phenotypes that we were interested in. And overall, this took about 16 months, and then we were able to take that data, hand it over to Energene, and they did their magic. And now Barak's going to explain a little bit about Energene and what they did with this data when we delivered it.
Okay, thank you, Chris. As mentioned by Chris, I'm going to start with a brief introduction to Energene, and then I'm going to describe some of the technical parts of the project. And so Energene is a genomics company. It was founded at 2010 with first line of products and services launched at 2015. And since then, we had over 280 projects around the world with 150 different customers. And you can check out our website for more information and over 25 peer-reviewed publications. So we provide computational tools and services to leading agribiotech companies, helping them to maximize their crop yield and significantly saving them time and cost. Let me see if I can. Okay, so my agenda for today, I'm going to briefly introduce the concepts of QDL mapping as we see it at Energene, the challenges, how we suggest the solution that we suggest, and then to see the results, we see the project design again, and then I'm going to show some of the results. So a brief introduction to how we see the genetic mapping problem. So usually when you think about genetic mapping, you think about finding genes that cause something. In many cases, they cause disease. In some cases, it's just a phenotype that you want to correct with the genotype, for example, height. But in breeding, you can think about it the other way around. Once you know something is genetic and you know what genes or you have markers for this in the genome, you can make predictions. And this has a lot of value in breeding. It can save a lot of time and cost. And so schematically, I can put the genetic mapping in 3 categories. In fact, there are 2 categories here. One of them is traditionally developed for pedigree based, and this is also the context of this project. So linkage analysis and QTL, they're based on pedigrees, and I will briefly discuss what this means. And GWAS is related to the population as a whole for unrelated samples. And so what are the advantages of working with a pedigree based? Well, the main advantage is that you control the diversity that you bring into the pedigree. This has a lot of advantages because you mask out a lot of the diversity in the population and a lot of noise. And also you can control to some extent, you can control the phenotypes. So you can make sure that within your pedigree, the phenotype is not rare, it's not extreme, or maybe it should be extreme. So the breeder can make his magic on the pedigree, and when he's satisfied, then you can choose the pedigree, the right pedigree for the project. And one of the advantages of working with plants, with plant genomes, is that you can generate many a large pedigree. So you can generate 100, 200, 300 samples within a given progeny, and this gives you a lot of statistical power. A basic workflow here at Energene for genetic mapping projects. We try to do things in an unbiased manner. So for each pedigree, we do an optimized marker discovery stage. When we have parents, we use the parents. We don't really need the parents for all the pedigree designs, but we always try to find the best markers for each pedigree. And then we scan the genome with those markers. And once we convince ourselves that we've detected QTLs, we build predictive model, which means that we generate markers that the breeders can use in the field to predict the phenotypes. And so to introduce the concept, so what are the markers that we use to run the QTL scans? I want to show you an example on progeny. This is not exactly the progeny used in this project, but it's not too far from there. Then I'm going to show the real progeny. So this is a classical four-way hybrid progeny. The parents are heterozygous and they're different. So each parent introduces two different alleles. Usually you don't, when you sequence the genome, you don't really know how to differentiate between the alleles, but theoretically you should be able to figure out what the four alleles are. So if you had a way of doing this, you could probably go to the progeny and see haplotype blocks such as in the diagram. So each haplotype block has a distinctive allele and it comes from a distinctive parent. And whenever we can, we try to infer those haplotype blocks and use them to do the statistical analysis, like with linkage analysis and GWAS and QTL analysis. And those haplotype blocks are highly informative. And I'm going to show you briefly how this is done and what are the challenges behind this. So if we focus on the maternal inheritance, just for simplicity, we know or we assume that we can represent the maternal genomic sequence with a reference genome. And this is the common concept. And behind this reference genome, you can get markers, sequence markers. In most cases, those are SNPs that would differentiate between parental and maternal lines. But this is not the end of the story because we know that the genome is a diploid and therefore markers should be able to differentiate also between the two alleles. And so our pipeline, what it tries to do is detect those SNPs, cluster them into the two alleles for each parent, and this stage is called phasing, and then infer the progeny haplotypes. And this is all done within a given pipeline with a set of algorithms that are optimized for next generation sequencing. Now, at the end of the day, if everything goes well, you get this view of the genome. So you know about the different alleles in the parental lines, you know about the haplotype blocks and how to distinguish between them on the progeny, and you have SNP markers or other sequence markers so that you can use it to validate the haplotype blocks. Now, how would you use haplotype blocks to do QTL or linkage analysis? Here's an illustration. For simplicity, I'm taking only a single parent. I'm calling this an F1, and this parent has two alleles, and the progeny is, you can see there are two groups in the progeny. So one of them is resistant to some pathogen and the other one is sensitive. And what you would like to do is you would like to find something in common, some haplotype block or a segment in the genome, which is in common to one group and not to the other group. Now, maybe you can see it, maybe not, but there is such a region right here. So all the resistant samples share this yellowish haplotype, while all the sensitive samples share this bluish haplotype. So this is a region of interest. The correlation between the phenotype and the genotype is quite obvious. And of course, we do this in a more rigorous manner using statistical tests. And usually we have a running score, such as the one you see above, indicating where are the interesting parts on the genome. So there are many advantages of working with haplotypes, even though inferring them is quite difficult. And so I want to emphasize two of them. One of them, and you'll see it in a minute, by inferring haplotype blocks, you reduce the number of markers that you use in QTL dramatically. And this has the advantage that you get a more accurate result, you have higher sensitivity, and you have better specificity, meaning that you do not have so many false positives, but you detect more. But this is not the end of the story, though. We can do this inference in the presence of missing data, and this is really important, and it's a big advantage because it means that we can go to the progeny now and take and sequence the progeny very shallow. So imagine you have 200 samples in the progeny and you need to sequence them. at 30x, that would have a high cost on this. But if you sequence them on 1x, then a project would become feasible. And this is the advantage. So we do a better job at a lower cost. And this is why we like this approach. Here's one way to look at the pipeline that we're running. So it's just it's a way to view what happens in the pipeline. And what I'm indicating here is the number of variants that we've detected and filtered along the way from the raw data all the way until we inferred the haplotype blocks. So you see, we start with 13 million different variants that are present in the parental lines, but we ended up with only 3,000 haplotype blocks, and we believe that this number is very close to reality. So you don't really need 13 million variants to represent 3,000 blocks. And much of our expertise and experience relies on this process of figuring out which of the sequence elements to use in order to infer the haplotypes correctly. And I would say as a side note here that with cannabis genomes, and that there are challenges that are so representing a cannabis genome with just one reference genome is particularly challenging. And what we started to do is we started to build fully phased genomes, as Chris said, and use this in order to figure out what are the markers to use that are most reliable. Right. And so before I'm gonna show the results, I'm gonna quickly show the pedigrees again. So the pedigree used here is a 6-way progeny. So it's more complicated because there are 6 different haplotypes segregated. But if I simplify this a little bit, this is the logic behind the progeny. So the autoflowering allele was introduced twice, and the hope was to see the autoflowering allele expressed as a homozygous in the autoflowering phenotype samples. And this is exactly what we saw before we started the sequencing. So we saw 25% of the samples with the autoflowering phenotype, and this was a really good starting point for the QTL mapping. So we knew we're gonna see a strong signal, and indeed, this is what we saw. So in this figure, I'm showing the chromosomes. They're encoded with letters, so it's a bit scrambled, but this is a real signal. And what you see here is the running score along the genome, and the significant level is right here, and is the black line right here. So the signal that we get for the autoflowering is huge. In fact, it's a perfect correlation, just about here. And it spans about 5 million base pairs, which is really, really nice. So it generated a lot of markers that Chris can use to validate and use in the field. And so this made everyone happy, of course, and this was an indication that everything went smooth and as expected. And when you think about such a project with 200 samples and DNA extraction and sequencing and all the stuff that could go wrong, so this was very nice to see. But I really want to conclude with different phenotypes. So in this project, Chris and his team also measured other phenotypes that had to do with the physical structure of the plants. And they did not know what to expect from this, and neither did we. But it turned out that almost all the phenotypes that they measured, except one maybe, all of them yielded a nice and significant signal in the genome. And so when we saw this, we knew there's so many things to discover here. This is just the beginning. And what we think is gonna happen, our vision for this, is that more and more signals are gonna come out like this, and they're gonna get incorporated in breeding programs. And then we're gonna start getting challenges that we're used to seeing in other crops that has, for example, trait correlations or interactions, or trait environment interactions, and other more complicated scenarios. And we're really looking forward to meeting these exciting challenges. That's it for me. Chris, you want to take it from here?
Okay, thank you, Barak. So the exciting part for us now is what do we do with these markers? And as a breeder, using these tools, enabling me to get to the market faster is very exciting. But first I want to explain kind of what we've been doing in a traditional approach without markers in autoflower breeding, and how we're going to then use those markers to be more innovative with our autoflower varieties. So in a traditional approach, we will take and make our initial cross to introduce the autoflower trait into our parent line that is photoperiod sensitive. So it has all the characteristics that we want, flower structure, trichome density, cannabinoid content, these things, but we want it to now be autoflower. So we make this initial cross to introduce the autoflower allele into, and we get the F1. Oh, went one too far. So in the F1, we will have the autoflower trait, which is recessive in a heterozygous state. So the F1 is photoperiod sensitive. So we have to then either SIB cross or self those F1 in order to get the autoflower to segregate. And then there's a few different things that we can do with the F2. We can either continue to do SIB crosses and improve the population and then release that as an OP variety once we're satisfied and have the traits fixed that we're interested in. Or we can self those F2s down and then use them in hybrid crosses, which is what we've been doing. Or we could take and back cross those F2s to the parent one. But the reason we haven't been doing this yet is because we've been waiting for the marker. Each back cross generation requires a validation step where we're either going to self or back cross to the autoflower parent to see which BC1 or back cross progeny have that allele in order to advance on. So it adds an extra step and it doubles your time for breeding. Whereas with the markers, we can take out that validation step because we do it with the markers. We don't have to do the actual crosses and go through a full cycle in the greenhouse. So that's how we'll be utilizing those markers. So we plan to use these markers in a back cross breeding program now. We'll incorporate this trait within 12 to 18 months into our breeding lines, which is very quick, and it enables us to get into the market quicker with new varieties. So here's just an overview of what this marker-assisted breeding program would look like. So we'll have our new population. We'll go through multiple cycles of marker-assisted selection. We'll develop our stable parent lines, and then we'll choose which parent lines from different populations to cross to create our stable hybrids. And then those will go into our trialing program that I mentioned earlier. And the way that the marker-assisted breeding step will look, there's a couple of approaches that we can take. So I'm just gonna walk you through those. The first step looks similar to the traditional. We just cross to the autoflower donor to get the F1. We then cross those F1 back to the recurrent parent to get the BC1, where we can then use the markers to identify which progeny carry the autoflower allele. We'd get rid of the ones that don't and advance the ones that do. And from this step, there's a couple ways we could go. We could either continue now selfing down the back cross one and get to our stable parent line. The problem with this is we might introduce a segment surrounding the autoflower allele that's larger than we want, which could bring in linkage drag, some traits that we don't want to incorporate, or those traits could be other places in the genome, as shown here at the bottom, that came from the autoflower parent. And some of those traits that we're not interested in carrying over are what you see in the picture at the top: the leafiness, low trichome density, the wrong bud shape, and lower cannabinoid content. So these are things that we want to address. And the way that we're going to address that is by continuing the backcross process to BC3 or BC5. So as we continue to cross back to that parent one, using the markers in each cycle to select those that still carry the allele, we can get to a backcross progeny that is both genetically and phenotypically very similar to our recurrent parent. And to kind of demonstrate what we're trying to get at, I included here some pictures from some of our trials. So starting at the left, you have a stable hybrid, but you can notice that it's fairly leafy. So this is okay if you're in the biomass market or even the pre-roll market, where those products are getting extracted or put into a cigarette. So the bud structure doesn't really matter much. And autoflowers can yield very high at the right planting densities in the field. So these are great. Growers that have been growing them for those markets are very happy. But the growers that want to grow an autoflower for the trimmed flower market, where the end consumer is getting essentially a bud, and that's the end product, autoflowers have typically fallen short. And I put some examples here on the right. So of what a good bud would look like. So as you can see with this top picture on the very right, a very high calyx to leaf ratio, easily trimmable. You get a nice round bud that can be packaged, and that's what the consumer is looking for. And in the autoflowers, we've been able to improve that without the marker through recurrent selection. As you can see in this third picture here from the left, we have achieved some higher calyx to leaf ratios, a little bit better of a bud structure, and this is acceptable, but it's not perfect. So with the markers, we're going to now be able to back cross, clean out those traits that we don't want from the autoflower parent, and have autoflowers that have buds that look like this on the right. And as a final slide, just wanted to give an example on where a breeding program can start and where it can get to. On the left, these are some of our commercial trials. This is a variety here on the left that hasn't gone through much breeding. It's okay for the biomass market or the pre-roll market, like I mentioned. It's highly variable. But if we put some work in, we clean up these varieties, we can develop stable parents, and we can end up with hybrids that produce fields that are uniform. The farmer can predict what they're going to get, and that's what's shown here on this middle picture. And then this is just a zoomed out view of our trials, commercial trials that we had going in California, one of many. And with that, we would like to say thank you guys for joining us, and I think now we'll take some questions.
Thank you, Chris and Brock, for this interesting talk. So we do have some time now from questions from the audience. The first question will be for Chris. You mentioned autoflowering plants being smaller than photoperiod dependent plants. How does that affect planting density and yield?
Yeah, so since the autoflower plants have a determinate growth and they flower within a certain period of time, not based on day length, they're typically shorter, with 2 to 3 feet in height. So you do have to plant them denser. But we've been doing trials with UC Riverside to look at planting density and see what densities are optimum. And we're finding once you get into planting densities over 20,000 plants per acre, which is far beyond what a lot of the photo period sensitive varieties are being planted at, you can get up to 4,000, 6,000 pounds to the acre of dried flower we've seen for biomass. So the yields do really go up if you're planting at the right planting densities with autoflowers.
Thank you, Chris. The next question is for Barack. What is the advantage of using sequence-based QTL mapping over using SNP arrays?
Okay, so SNP arrays and genotyping platform are good stable technology, and we'd like to use them when we can. The problem is they're not always optimized for all the species. For example, for the cannabis, it's a highly diverse species, and we see it even in our own project. It's kind of difficult to find common genotypes for many varieties. So you end up having a lot of SNPs that you cannot use. They're not well distributed, and eventually you have underperformance. So we'd like to use the sequencing base, which is unbiased, and optimize the marker discovery per pedigree. So it might cost a little bit more, but it's optimized, and we think that it's a better solution at this state. There might be, we can imagine a platform, a set of SNPs that might represent the entire population, but we don't know that yet. We need to do a diversity analysis for this, and this hasn't been done yet. So we prefer at the moment to work in an unbiased manner.
Thank you, Barack. This next one is also for you. Have you been able to identify the autoflowering gene?
So no, we haven't really tried, because the main goal for QDL project is to find markers, and this has immediate implication for the breeding programs. Finding the genes, it could be done as a separate project, but it could be also very risky, as there are many variations and many mutations in the genome, and you can, it's kind of difficult to figure out what is the causative gene within a 5 million base per confidence interval. But I can imagine that with time, yes, it could be done, but it would require a different type of effort.
Our next question is for Chris. Do you have high CBD varieties that are over 20% CBD and less than 3% THC?
So this is a difficult task to accomplish. What we've learned through our breeding program is that as you approach, and Larry Smart did some of this work too over at Cornell, as you approach 10%, even like 8 to 10%, the THC will start to creep over that 0.3%. So far, I haven't seen anybody that has broke that relationship. We've seen some instances where we do test a cannabis variety, a hemp variety that is over the 10% and is below 0.3. So we think that it might be possible to break that, but we have to take things further and really test it across multiple environments and see how that plays out.
The next question is, what marker system are you using? And how many markers do you have linked to the day neutral gene? And this question is directed to Barack.
Okay, so I'm not sure that I understand what is a marker system, but I'm going to answer what I do understand. So we're trying to give Chris markers that are actually SNPs, and they're pretty isolated and with a nice sequence content, because what we imagine Chris would do is he would design PCR on those and try to use them in the field. So the markers themselves in this particular projects are SNPs, and they have nice, they're nice to PCR. You can PCR them. And I don't remember the number of SNPs that are linked to this, but it's of the order of at least 100 markers. And I guess Chris doesn't need so many markers, so he can choose whatever works well in the field.
The next question goes back to Chris. Is your marker effective with other day-neutral pedigrees, such as Finola or Oregon CBD pedigrees?
So this we don't know yet. We just got the markers. We have to go through our validation period. We'll choose from the markers, like Barack said. We have quite a few to choose from. We'll put those through an assay, see which ones work best within our own germplasm. And then the idea will be to look at optimizing that across populations that are not at all related to the autoflower donor that we used for the mapping, and see if it is conserved across different autoflower varieties besides the one that we used. So we don't know yet, but we should know here within the next year or so.
For Barack, does your pipeline take into consideration structural variation?
Okay, that's a tricky question. The answer is yes and no. I mean, yes, it is that we can infer the structural variation easily with the data that we have. But the thing is that we don't really know what to do with structural variations. In fact, if we do not, if we cannot correlate between structural variations themselves and the phenotype, then these structural variations have no meaning in the project. So the answer is, in this project, there are structural variations, but they are not participating in the QTL signal. So the explanation is pretty simple. We don't report on them.
Chris, what experiences from your watermelon and wheat breeding are most helpful to your current efforts?
So within the projects that I did with watermelon and within wheat, I got a lot of experience in using markers in a commercial breeding program. So being involved in a commercial program at Syngenta for watermelon, I got to work with markers, a diverse team, and understand breeding at a large scale for a global market. And those have come in handy in approaching in hemp and cannabis.
This one's also for you, Chris. How will you maintain the superior autoflower strain for next generation?
I'm not sure exactly what that means, but the way that we create a variety is that we'll develop two parent lines from different populations, and then those are, once they're stable, so they'll breed true for their traits. Those parent lines are continuously self pollinated, so we can maintain them that way. And then to create a variety, we cross one parent with another parent, which creates the F1 hybrid, and that will always be the same so long as we're always using the same parents. So I hope that answers the question. I think that's maybe what they were getting at with that question.
Thank you. And this one is also for you. Chris, are you able to screen certain terpenes, olfactory parameters, and bring this trait into next generation like you do with the autoflower trait?
Yes, this is something that we're interested in doing in the future. We haven't done yet, but if it segregates within cannabis, then we can map it, as long as we can also phenotype it very accurately.
And this next one is for Barack. From your QTL plot, you show you have a resolution of an entire chromosome. How is that possible?
Okay, so I don't really understand what are the question. So the way that we so I'm not sure that I understand. So the way that we extract markers, we extract marker uniformly on the entire chromosome. So we see the entire chromosome. and we can calculate the statistical significance of each of the locations on the chromosome. Maybe the question is, how come the entire chromosome is linked to the autoflowering? Maybe this was the question. I'm not sure.
We just got feedback. No, I mean the QTL that you show is as large as a chromosome.
Yes. Okay. So this is what I thought. So it looks as if the entire chromosome is linked, and this is because what you see is you see a perfect correlation for a categorical variable. So in fact, this is not QTL, it's linkage analysis, and you don't need 200 samples to link this. So this is just indicating that the signal is so high that the entire chromosome seems to be linked. But if you look closely on the figure, I don't know if you can show the figure, actually there's a different, if you can go back, no, go forward, yeah, another one, yeah. So actually there's a something that looks like a very small hill around the peak. It's not small at all, it's very, very large. And so the fact is that you have a perfect correlation for 200 samples, so everything goes highly, highly significant. But in fact is that the small peak that you see there, which looks small, it's very, 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 very large. And this is because the sample size is very large. And so this is just a manifestation of that we're dealing with a recessive trait with 200 samples, which is a huge sample size for this. And we could have, you know, we could have seen this with a much lower resolution, a much lower sample size, and things would go down a little bit. So it's, I agree that this is confusing, but it's not an artifact.
Thank you, Barak and Chris, and thank you to the audience for all your questions. But we are unfortunately running out of time, and I'm sorry we couldn't answer all of your questions, but please note that we will follow up by email to answer them all after the webinar. I'd like to again thank Chris and Barak for their time today. Thank you for sharing with us your presentations. This webinar was recorded and we'll have it available in the next few days, so keep an eye out for our email with a link to the recording. We will also have it up on our website. If you have further questions or an interesting research project you'd like to discuss with our experts, please don't hesitate to contact us at nrgene.com. Thank you again for joining us today and enjoy the rest of your day.